Hello students, today we will start the next part of strategies for enhancement in food production, and today we will study the applications of plant breeding. In last lecture you have studied that the various that is steps involved in the plant breeding, and now today we will discuss the applications of the plant breeding for disease resistance. For example, some disease resistant varieties of crop and insect resistance also, example of some insect resistant varieties of crops, and improved food quality like biofortification, and some of the biofortified crops are there. Next, that part tissue culture and its technique, this we will discuss in the next lecture. Today we will study this also, single cell protein, its source and significance. So only this micropropagation, this is a lengthy method that we are going to study in the next part. So applications of plant breeding. So you know that the new varieties developed in India by the plant breeding, and crop plants are often affected by the bacterial and fungal pathogens, and these infections on the one hand reduce the quality of the product, and on the other hand significantly reduce the quantity of the product also. So they affect the quality as well as quantity of the agricultural product. So some of the varieties of common crops which have been generated by plant breeding techniques in order to make them resistant to the different types of diseases. So let me see what are these crops. So mainly as per the syllabus, these are the crops and their varieties you have to learn, and for which the diseases they are resistant. So the first that crop is wheat, variety Himgiri. It is resistant to leaf and stripe rust. Brassica, its variety Pusa Swarnim. It is resistant to white rust. Then cauliflower, its varieties are there Pusa Shubhra, Pusa Snowball, and it is resistant to black rot and curl blight. And cowpea, its variety Pusa Komal, and it is resistant to the bacterial blight. Then chili, its variety Pusa Sadabahar, it is resistant to chili mosaic virus, tobacco mosaic virus, and leaf curl. So these are the varieties of the crop developed by the plant breeding techniques which are resistant to these different diseases. Now another aspect, that some varieties which are resistant to the insect pests. So pests are these insects which feed on the different parts of the plant and do not allow them to develop properly. So what are these crops? They are Brassica, that is mustard, its variety Pusa Gaurav, and it is resistant to the aphids. Flat bean Pusa Sem 2, Pusa Sem 3, and it is resistant to jacids, aphids, and fruit borer. Then okra, its variety Pusa Savani, Pusa A4, and insects to which it is resistant, they are shoot and fruit borer. So these all are the varieties of the crop, and they are resistant to all these insect or pests. So understood, children? So these are the different varieties of the crops developed by the plant breeding techniques. Now next we will study the plant breeding for improved nutritional quality, because more than 15% of the world's population does not have sufficient amount of food to meet their daily nutrients, or the daily nutritional requirement. Many of them, they are getting food of poor quality which has insufficient protein and vitamins. So the condition is worst in developing countries of Africa and Asia. So plant breeding is the technique by which such type of crops can be developed which are rich in vitamins and the essential nutrients. So one of the technique we call it as the biofortification. Biofortification is a process of increasing the density of vitamins and minerals in a crop through plant breeding or agronomic practices, so that biofortified crops, when consumed regularly, will generate measurable improvement in vitamin and mineral nutritional status. Means biofortification is the process of breeding food crops that are rich in biologically important micronutrients. These crops fortify themselves and they load high levels of minerals and vitamins in their seeds and roots, which are then they are harvested and then eaten. So through biofortification, scientists can provide farmers with crop varieties that naturally reduce anemia, cognitive impairment, and other nutritionally related health problems, potentially in hundreds of millions of people. Means these crops are rich in particular vitamin and the minerals, and the person, those who have deficiency of this particular mineral or the vitamin, they can consume these crops or the food from these crops, and that amount of deficiency can be overcome. So let me see here that biofortification is the process of breeding crops with higher levels of vitamins and minerals, or higher protein and healthier fats. Breeding for improved nutritional quality is undertaken with the objective of improving. Means what are the objective of the biofortification? Means here increasing protein content and quality, increasing oil content and quality, and increasing vitamin content, and increasing micronutrient and mineral content. So these are the objectives of the biofortification, to increase all these that content in the crop. For examples, vitamin A enriched carrots, spinach, pumpkin; vitamin C enriched bitter gourd, mustard, tomato; iron and calcium enriched spinach. So these are the examples of such crops. Now, till now we have developed many hybrid fortified varieties. So in India also and in other country also, these biofortified crops are developed. For example, that crop sweet potato, it is rich in provitamin A, which is released in the countries, means countries of first release, these are the Mozambique and Uganda in 2007. Beans, they are rich in iron and zinc. Pearl millet, they are also rich in iron and zinc, and they are released in India in 2013. Cassava, which is rich in provitamin A, released in Nigeria in 2013. Maize, provitamin A, rich in provitamin A, and rice, which is rich in zinc and iron, and it is released in Bangladesh and India in 2013. Wheat, which is also rich in zinc and iron, and countries of release are India and Pakistan, 2013. And in 2000, a hybrid variety of maize also was developed, which has twice the content of lysine and tryptophan compared to the earlier hybrid maize varieties. Atlas 66, it is a wheat variety which is rich in protein content and has been used as a donor for improving cultivated wheat. So we have developed many varieties of rice and tomatoes fortified with the iron. So all these are the examples of biofortified crops. Now next we will study the single cell protein. So another aspect in which technology is used, that is the single cell protein. So a digestible protein extracted from a microbe in a culture medium, either cultured algae, yeast or bacteria. This is the single cell protein. So what is the single cell protein? The term single cell protein refers to the total protein extracted from the pure culture of microorganisms. For example, yeast, algae, filamentous fungi and bacteria. This SCPs are dried cells of microorganisms which can be used as dietary protein supplement. They are used as animal feed and can be used for human feed as protein supplement, also called novel food or mini food. It is also known as microbial protein because we are getting it from the microorganisms. 60 to 80% dry cell weight contains nucleic acid, fats, carbohydrates, vitamins and minerals. Now, single cell protein can be produced from all these microorganisms we have discussed already. Some low cost substrates are used to produce microbial biomass to produce single cell proteins. SCP is rich in high quality protein and is low in fat content, hence it is a desirable human food. SCP should also reduce the pressure on agricultural production systems for the supply of proteins, and it can reduce environmental pollution. Now, examples. For example, microbes like spirulina. It can be grown easily on materials like wastewater from potato processing plants, straw, molasses, animal manure and even sewage to produce large quantities and can serve as food rich in protein, minerals, fats, carbohydrates and vitamins. So, this is blue-green algae spirulina. Some common microbes as SCP producers, example cyanobacteria like spirulina, bacteria like Methylophilus methylotrophus, yeast Candida utilis, filamentous fungi Fusarium gramiarum. So these are some microbes used as SCP. Advantages of SCP: it is rich in high quality protein and poor in fat content. It reduces the pressure on agricultural production systems for the supply of the required protein. SCP production is based on industrial effluents, so it helps to minimize environmental pollution. Like just now explained you that these microorganisms can be grown on the industrial effluents, the waste. SCP can be produced in laboratory throughout the year, means they don't need the particular that season, climate or land or soil or fertilizer. So they can be produced in laboratories throughout the year. So all these are the advantages of single cell protein. So students, today we have studied the application of the plant breeding and certain other ways through which food can be produced. So in the next that part we will study the micropropagation or tissue culture.